Cell Respiration by Briana Mercury, Fred Canas, and Maegan Madrigal. The background information for this lab is that it is similar to photosynthesis. The sugars are our main source of cell energy. Energy stored in is ATP that can become ADP, constant cycle glycolysis, acetyl CoA synthesis, Karen cycle, and ETC. Glycolysis 10 steps to break down glucose to pyruvate, no net gain of ATP. Pyruvate converted to acetyl CoA that is then broken down in Krebs cycle. Electrons and electron carriers used to make additional ATP through ETC. ADH and FADH2 is used in ETC to generate more ATP. As the electrons pass, they release energy used to shuttle hydrogen ions. ATP synthase as a phosphate to ADP. ATP made altogether. Oh, six. D6. What number is this, Megan? 36 ATP made altogether. I'm very sorry. Respirometer to measure rate of respiration of germinating and dormant.
That was beautiful.
Now we have our pre-lab questions. Pre-lab question number 1 was, during respiration, a seed metabolizes sugars. What is the source of the sugar metabolized by the seed? And our answer was that it was metabolized from the parents. That is the source. The second question, what variable do you think affect the respiration rate of the seeds? We said that the gas in the air, the activity, the weight, the elevation, and the temperature. Lab questions. So our hypothesis for this experiment is that the germinating peas will respirate. Our independent variable is time, and the dependent variable is volume. And here we have a volume versus time graph, and it just shows all of our data here. Then we have all the variables that we had, the controls we had. So the volume inside was a control because it stayed the same. The amount of KOH was the same for all of them. The time and the temperature was also the same. Another question, which of the respirometers serves as a control? And we said 3 because it was just beads, so it shouldn't have been breathing since it's not a living thing. Number 5, assuming your control measures worked, tell what variables change to bring about the observed change in volume. And we said it's volume and moles. Using your graph and data tables, summarize your findings comparing results from respirometer 1 and 2. We said that respirometer 1 has more than respirometer 2 because respirometer 1 is germinated. So it breathes more, we think. Actually, no, using our data. Germinating seeds at room temperature had 0.095 milliliters a minute. That was the rate of oxygen consumed. And the dormant seeds at room temperature were 0.005. So that proves what we just said in number 6. Part 2, temperature in peas. So this was our idea for the lab experiment after we did the one that I just talked about. So with this one, our question was, will the temperature affect respiration rates? Our hypothesis was that we believe that in more heat, the peas will respirate faster. I didn't agree with Megan on this one. I thought the cold ones would, but she said that we had to do heat, so whatever. Variables and controls. Independent variable is time, and the dependent variable is temperature because we will be changing it from cold, hot, and the average temperature like we did in the first experiment. We're just using that. Then our procedure. We're going to follow the same procedure as we did before, but we will not have one tray at room temperature because we already have that information. Instead, we will have 2 trays, one in the fridge at about 60 degrees Fahrenheit and one on a hot plate that exceeded 120 degrees Fahrenheit. Our analysis. Beautiful. So we have, starting at a time of 0, we had 6, 6, and 7. Here is the germinating peas. This is the peas and beads, so the dormant peas, and then just plain beads. So this is all our information. At one point this is actually just the cold, so this is in the fridge. At one point we couldn't see anything. Then this is our analysis continued. This is the same stuff, germinating peas, peas with beads, and just beads. But in the hot temperature, all our information right here. At each time, the amount of air. So in conclusion, we concluded that the peas respirated better in the refrigerator under the temperatures of 56 to 58 degrees Fahrenheit. So Briana was right, not Maegan. In the heat at temperatures of 120 degrees and above, the peas barely respirated at all.